Secondary electrospray ionization mass spectrometry, or SESI-MS, enables the analysis of bacterial volatiles without the requirement of any sample pretreatment. The headspace of the bacterial culture is displaced by carbon dioxide into the SESI reaction chamber. As the volatiles traverse the SESI reaction chamber, they pass through the electrospray cloud and become ionized. Once ionized, the volatiles are pulled into the mass spectrometer for analysis. Excess carrier gas and unreacted bacterial volatiles are passed through a 0.22 micron filter to remove any bacterial agents as an additional measure of protection and vented to a chemical hood. Ultimately, this SESI-MS protocol offers bacterial volatile fingerprints within minutes.
We anticipate that this technology could be used in many applications, including exhale breath analysis for infectious diseases, or even applied to food systems where we might be able to rapidly detect pathogens that are on or in our food.
There are several main advantages to this technique over existing methods like GC-MS, SIFT-MS, and PTR-MS. First, the volatiles don't require any pretreatment prior to analysis. Second, it's possible to fragment specific ions in the volatile mixture, which makes compound identification easier. And third, it's rapid. It typically takes less than 5 minutes to analyze each sample.
Choose the appropriate vessel for growing your culture, considering the growth requirements of the species as well as the efficient delivery of volatiles to the mass spectrometer. In this demonstration, we have selected standard 100 milliliter Pyrex media bottles. The bacterial volatiles are delivered to the mass spectrometer by a stream of carrier gas. To assemble the